So Frederick Sanger had developed a method for sequencing amino acid in protein. So how you can sequence amino acid in protein? Amino acids, from where you get amino acid? You remember central dogma? From DNA you get RNA. From RNA you get protein. What is protein made up of? Amine. That is what is that carboxylic and amine together called? Amino acid. Amino group and acid group.
The one you're learning in
Yeah, you are learning chemistry. On one side NH2, the other side COH. That's what amino acid it is called. Okay. So if you know the amino acid, can you find the codons? Here you know AUG, that three triplet. One particular codon codes for a particular amino acid. So if you know amino acid, you can find out codon. Similarly, same method they use for sequencing the DNA. If you can sequence amino acid, you can know the sequence of the RNA. So same method they use for sequencing the DNA. So DNA is converted into random fragments. Okay. So again, they are cutting the DNA into small pieces because it is almost 2.2 meter long and it is very difficult. It will take lot of time to study the entire DNA at one stretch. That's why they cut the DNA into many pieces and study them separately and then again join them. Okay. And there is a technical limitations in sequencing very long pieces of DNA. So there are some limitations. You cannot sequence a long stretch of DNA. So you cut the DNA into many pieces, study it, and then join it. HGP was closely associated with bioinformatics. Okay, bioinformatics, computer and biology together, bioinformatics. Bioinformatics, application of computer science and information technology to the field of biology and medicine. Okay, so you have copied this bioinformatics definition in Human Genome Project. This you all know. Out of the 24 chromosome, 23 chromosome. Okay. Of the 23 chromosome, this is not 24, 23 chromosome, 22 autosomes and X and Y chromosome. Yeah, it is 24 chromosome. Okay. Out of 20, why they have mentioned 24 chromosome? X and Y are not same chromosome. X is different, Y is different. 22 are autosome and there is one X and one Y chromosome. The last sequenced one is chromosome 1, May 2006. Okay. Genetic and physical maps of the genome were generated using information on polymorphism of restriction endonuclease, recognition side and some repetitive DNA sequence microsatellite. So restriction endonuclease and repetitive DNA sequence, it is in detail in next slides. What is restriction endonuclease? Endonuclease are enzyme which cut the DNA. Okay, now you have a long stretch of DNA, you have to cut the DNA. So for that cutting, they were using restriction endonuclease. Now there are two types of nuclease enzyme, endo and exo. Endo will cut the DNA in the center, somewhere in the center. Exonuclease will cut the DNA at the edges. DNA sequencing has also been done in bacteria, yeast, and C. elegans. C. elegans is nematode. Drosophila, what is the another name for Drosophila? Fruit fly. Okay. Plants like rice and Arabidopsis. This is the procedure. Take the organ or tissue, from that extract DNA. Okay. Then cut the DNA fragments. Okay. Then single sequence, single fragment, and make many copies and then put under the computer and do studies. Everything is computerized. Okay. Everything will be computerized. This is PCR. PCR for polymerase chain reaction. Okay. That full form is asked. It will come later. Okay. Salient features of human genome. Human genome contains 3164.7 million nucleotide bases. 3164.7 million nucleotide bases. What was the cost for one base pair? $3. So almost 9,3 somewhere 64 3s are 182. So 9492 something $1 million the cost. Okay. Total number of genes is about 30,000. Okay. Average gene consists of 3,000 bases, but the size varies. Largest known human gene is dystrophin on X chromosome and contains 2.4 million base pairs. This dystrophin is a protein which is related to muscle. Dystrophin. Now, if there is some mutation, then you get muscle dystrophy, where the muscle start destroying by itself. Muscle get rotten, like parts. Leprosy, you all know, no? Finger cutting, that is it. Okay. So then salient features of human genome. 99.9 nucleotide is same. In all the human, 99.9% of the nucleotide is same. So all will have almost 99.9% human same sequences. Only 0.1% is different, which makes every individual unique. Only 0.1 difference is there in the DNA. Functions of over 50 discovered genes are known. 50% of the discovered genes which are found, the function is known. Chromosome 1 has the most gene, that is 2968, and chromosome Y have the fewest. Less than 2% of the genome codes for protein. Only less than 2%. Rest all are introns or UTR, untranslated region. Only 2% code for protein. Very large portion. Now this is important. Salient feature of human genome. Now this is that repeated sequences. Very large portion of human genome is made up of repeated sequences. Very large portion. Most of the sequences are repeated and these do not code for protein. Okay. These are stretches of DNA that are repeated many times. They have no direct coding function. They just help in giving chromosome structure, dynamic, stability, and evolution. So these repetitive sequences help to increase the size of the chromosome, or it can help to make the chromosome stable, and also it tells about evolution. I told these sequences which are not working in us were working in our ancestors. So that tells you about evolution. About 1.4 million locations have been base pair differences. 1.4 million locations have single base pair differences. Single, one base pair will be changed. This is called as SNP, small nucleotide polymorphism, or SNPs. This helps to find chromosome locations for disease-associated sequences and tracing human history. Okay. This is that repeats. A person 1 will have 8 repeats, person 2 can have 9 repeats, person 3 can have 10 repeats. Okay. So these are unique for particular person. Everyone will be different based on these sequences. Now to find your DNA and your DNA to any difference, what they will try to find is this repeats. They will find whether these repeats are there or not. Rest all will be same. Then DNA fingerprinting. So before this, you all just write whatever is important.